Thank you everyone for joining us at the InVivaScribe Symposium titled Unlocking the Potential of NGS Clonality and MRD Testing from Research to High-Throughput Environments. I'm honored to introduce our first speaker today, Dr. Artur Kibler from the Institute of Cell Biology at the University of Duisburg-Essen. Dr. Kibler will be presenting on the topic Beyond Systemic Memory B-Cell Archiving, Mining B-Cell Receptor Repertoires for Antigen-Specific Clones and Tumor Cell Precursors. Dr. Kibler started his PhD in the lab of Professor Ralf Küppers at the University of Duisburg-Essen in 2016. In his PhD thesis, he focused on the functional and molecular changes of human splenic marginal zone B cells throughout the human lifespan. He finished his PhD in 2021 with summa cum laude, and subsequently, he received the BioMe Postdoctoral Excellence Program grant. Currently, his research focuses on age-associated changes in the SMZ microenvironment and B-cell myeloid cell crosstalk. In the autumn of this year, he will join the group of Professor Daniel Engel, who focuses on the immune responses during acute bacterial infections.
Thank you for the kind introduction. So today I'm going to talk about the molecular analysis of B-cell receptor repertoires and what we can learn from it about the splenic immunity and also for lymphomagenesis. So this is my conflict of interest. First of all, the spleen is important in defense against infections of the bloodstream because the spleen is the largest filter of the peripheral blood in the human body. And in particular, the splenic marginal zone and the immune cells inhabited there are important for this function. As you may know, the spleen is really important in the protection against infections with encapsulated bacteria. However, we know that also people who have a spleen are at higher risk because of their age. In particular, neonates and infants, as well as seniors, are at elevated risk of suffering from severe infections with, for example, streptococcus pneumonia as displayed in this graph. And also in these age groups, the incidence of severe complications such as sepsis or meningitis is increased. However, the molecular reasons for this increased susceptibility is yet unknown. Here you can see immunofluorescence staining of the human splenic marginal zone. In the first graph in the upper left corner, there's the staining of CD21. And there, the pink outline highlights the different areas. The high expression in the center marks the follicular dendritic cells in the B-cell follicle, which are important during germinal center reactions. And the more faint staining at the periphery of the B-cell follicle, this is the marginal zone. As you can see from the other stainings, these marginal zone B cells are characterized by expression of IgM, and the vast majority of these cells also expresses CD27, the human memory B cell markers. These cells are characterized by an increased cell size, and they are easily activatable. So in response to any foreign pathogen, they quickly differentiate to plasma cells and secrete large amounts of immunoglobulin into the circulation. We had many open questions to human splenic marginal zone B cells because this compartment is very good analyzed in mice. However, many of these questions were not addressed in the human situation yet. We had different experimental strategies to address these questions, and to our surprise, many of these questions could be addressed by simply analyzing the B-cell receptor repertoire. First of all, a bit of data regarding our cohort. So in total, we analyzed over 190 individuals. Among these were 150 spleens. A large fraction of them were also analyzed in parallel with paired peripheral blood from the same individual. And this cohort was complemented by 40 age and gender matched peripheral blood samples. Here you can see our gating strategy to distinguish the different B-cell subsets that are present in the spleen. maybe it's not so important to go into all the details here. What you can learn from this slide is that we differentiated up to 8 different subsets from peripheral blood and spleen. And the main difference you can see in the upper left corner, the first graph, there's this CD21 high subset in spleen, which is absent in blood. These are the splenic marginal zone B cells, as defined by flow cytometry. Here you can see that throughout life, the composition of splenic and peripheral blood B cell subsets changes substantially. So for one thing, obviously early in life, you do not have so many memory B cells and they slowly accumulate. And also what you can see here is that spleen and peripheral blood differ substantially. So the peripheral blood is dominated by a high frequency of naive B cells, which are important in new immune responses against yet unknown pathogens, whereas memory B cells are important in recall responses. Here, again, you can see the major difference between spleen and blood, so the presence of the marginal zone B cell compartment, which is gated here in the upper row. And by a combination of IgM and CD21, we can distinguish between conventional IgM positive marginal zone B cells, as they are, for example, characterized in mice, and which is which is a large difference to mice. There's also an additional compartment of CD21 high cells expressing either IgG or IgA. So this is a class-switched compartment of marginal zone B cells. These cells obviously have more past germinal center experience. And as you can see throughout life, the composition of the marginal zone changes, while the IgM expressing marginal zone B cells remain at a quite constant level, around 10 to 15% throughout life. In the 2-month year-old individual, you can see that there are hardly any class-switched marginal zone B cells. And they slowly accumulate until late childhood or early adulthood. They reach a similar abundance as the IgM-expressing marginal zone B cells, around 10 to 15%. However, in the very old individual that you can see here on the far right, in the 74-year-old, the class-switched marginal zone B cells dominate the splenic B cell pool. Here we quantified the abundance of these 2 marginal zone B cell subsets in all our 150 individuals. And as you can see, quite strikingly, on the left-hand side, there's no major change in the abundance of marginal zone B cells expressing the IgM isotype, while on the right-hand side, the class-switched marginal zone B cells are virtually absent at birth and only slowly accumulate after the age of 2 to 4 years. And there's another interesting observation that we made. Around 40 to 60 years, we see that there is a fraction of individuals which has quite a high abundance of these cells. So even though the epidemiological risk of infection, for example, Streptococcus pneumoniae, is not elevated yet, in these people we see that this compartment of class-switched marginal zone B cells is increased, which might influence the ability of the spleen to respond to yet unknown pathogens. Here you can see a phenotypic analysis of the B cell subsets from peripheral blood and spleen that I introduced earlier. As you can see, there are additional markers that distinguish conventional memory B cells and marginal zone B cell subsets, both in spleen and blood. Interestingly, the expression of the receptors CD21 and CD35, which you might know are the complement receptors 2 and 1, are dramatically increased in these cells compared to the conventional naive and memory B cells from spleen. They are very important in the biology of marginal zone B cells when it comes to infections with encapsulated bacteria. It is well established that complement components are important for the recognition of these bacteria and their uptake. And in particular, marginal zone B cells have a high expression level of these molecules. In addition, the molecule CD148 is elevated in, especially in the IgM-expressing marginal zone B cells. This molecule is similar to CD45 and mediates quiescence of these cells. Next, so basically what we saw here is that marginal zone B cells differ phenotypically. Next, we investigated the functional differences of these cells. And as you can see here, this is an analysis of cellular proliferation upon stimulation. And these analyses were performed on the same day. And as you can see on the right, marginal zone B cells show a higher proliferation capacity compared to both spleen and blood-derived conventional memory B cells, irrespective of the stimulation condition. In the upper row, you can see the proliferation capacity in response to the TLR9 ligand CpG. And in the lower row, we used a typical stimulation condition which is used for B cells, consisting of a stimulus that triggers the B cell receptor signaling in combination with CD40 ligand, the main signal that B cells receive from T cells, for example, during the germinal center reaction. Here is a summary of these proliferation experiments. And as you can see, for example, in the TD stimulation on after 48 hours, so in the lower left panel, marginal zone B cells and class-switch memory B cells from spleen start proliferation approximately 1 day earlier compared to conventional B cell subsets from peripheral blood. And also the response, the proliferation response to CpG stimulation is quite unique to the marginal zone. The other B cells really lag behind in these conditions. So in summary, from this first part, I hope I could convince you that these B cell subsets that we investigate here are both phenotypically and functionally different. And that the composition of the B cell compartment, both in spleen and blood, changes throughout life, and these changes might contribute to the increased susceptibility to infection in the risk groups. Next, I will focus on the B cell receptor deep sequencing analysis that we performed. For this analysis, we selected 17 donors in the age ranging from 0 to 77 years. And from these 17 individuals, we had paired peripheral blood and spleen biopsies available. We used the next generation sequencing kit of InVivoScribe, which amplifies the heavy chains, including the framework region 1. And the memory B cells that were analyzed in these experiments were isolated by FACS sorting. First, a short introduction. So maybe you are aware, but anyway, what is characteristic for a B cell is its unique B cell receptor, which is generated in the B cell development in the bone marrow. And the B cell receptor is produced in a process which is called VDJ recombination. From the germline encoded genes, V, D, and J genes, a random pair of D and J genes is first rearranged, and then a V gene is linked to this successfully rearranged product. And at the linkage sites, there is a mechanism which introduces additional heterogeneity. So there are nucleotides either introduced or removed in a random fashion, which increases the heterogeneity. So even if you would analyze two B cells which have the identical rearrangement of V, D, and J genes, this increased heterogeneity at the linkage sites leads to unique sequences in these two B cells. And then the final rearrangement obviously also has a constant region, which can either be, we mainly focus on IgM, IgG, and IgA because of their abundance and importance in immune responses. So this is basically our workflow. As I stated earlier, we had paired samples available from spleen and blood and isolated the B cells by first using MUX and then FACS sorting, isolated the DNA, and then the unique V, D, and J sequences of each B cell were amplified from the DNA, which is important to distinguish the PCR amplifications from unique sequences in the end. Using the in vivo scribe kit, we amplified these VDJ sequences, including the important CDR3 region. And then basically we sequenced our library. And next you need to know, after the B cell is generated, so after the B cell generates its B cell receptor in the VDJ recombination, upon activation of a naive B cell, there are several additional pathways which can contribute to higher diversity of the B cell receptor repertoire. And these pathways include, first of all, clonal expansion, which is basically just proliferation, but also somatic hypermutation and class switch recombination. These two processes occur during immune responses in the germinal center and lead to additional point mutations in the B cell receptor gene, as well as exchange of the constant region from IgM, for example, to IgG or IgA. Our definition of clonal lineages was the, first of all, we collapsed all identical sequences that were generated in the sequencing because of PCR amplification, and collapsed them to the single longest detected IgHV sequence, which was present in each vial. And then we defined our clones by using this identical IgA-heavy gene, as well as 95% homology and up to 5% variance in sequence length. And clones were only defined if the sequences were present in separate vials. So first of all, this is a basic analysis of the median mutation frequency of the IgA-heavy genes. And as you can see, which is expected, in small children, the mutation frequency is quite low. These individuals lack a immune history, and it is only built up over time. However, what you can also see is that we reach a plateau roughly between 10 and 20 years. After that, the median mutation frequency doesn't change so much in the memory compartments. However, IgM and class-switched compartments differ in their median mutation frequency, as you can see. While the frequency is around 4% for the IgM compartment, it's roughly 6 to 8% for the class-switched memory B cell subsets, both in spleen and blood. Here you can see a clonality analysis. So basically the blue area in these spider plots represents the fraction of sequences that correspond to clones. While in infants, we don't see many clones. This changes quite early during childhood. Already in the 4-year-old, virtually all sequences from peripheral blood belong to expanded clones, while only a small fraction of splenic clones belongs to clones. This changes around 40 to 50 years. And in the very old individuals, here 69 and 77 years old, we see that approximately 80% of all sequences that we analyzed, also in spleen, belong to expanded clones. What you can see here is also a clonality analysis, but this time from the perspective of tissues. So the ribbons represent sequences that are shared in clones between blood and spleen. And as you can see, the grayish area of the peripheral blood sequences is almost completely covered by ribbons. So that means that we, whenever we retrieve a sequence from peripheral blood, we find clonally related sequences in spleen. The other way around, in spleen, the vast majority of sequences is unique, at least in in this younger donor. If we consider now more individual donors, the picture is quite similar. So we see that all the peripheral blood derived sequences correspond to shared clones between blood and spleen. And this leads us to the conclusion that the spleen has the important function to systematically archive circulating memory B cells throughout life. So whenever a memory B cell is generated elsewhere in the body, for example in the tonsils or lymph nodes throughout the body, and it reaches the bloodstream, members of these clones eventually end up in the spleen and are integrated into this archive. However, this human memory B cell archive in the spleen changes with age. So what we observed first is that the average clone size increases throughout life, and while the fraction of shared clones is quite constant throughout life, the fraction of clones that are exclusively present in the spleen increases around 40 to 60 years of age. So you could summarize these findings in the following way. While already early in life, a major fraction of splenic peripheral blood-derived sequences corresponds to splenic clones, so shared clones with the spleen. There's a high fraction of unique sequences in the spleen. So there's a high heterogeneity, which makes the splenic immune system flexible, so we think. While during aging, the fraction of unique sequences in the spleen is severely diminished, and most sequences belong either to shared or to splenic clones. You can see examples of B-cell receptor dendrograms. So basically, this is just a visualization of sequences that belong to one single clone. And the further down one of the colored circles in these plots is the higher the Ig mutation frequency. And as you can see in these two examples, we saw a quite reminiscent feature of these shared clones. Most sequences that were derived from blood correspond to one single diversified branch in these dendrograms. And when we quantified this feature, we saw that there is a peak early in life. We think the two lower points here are due to low Ig mutation frequency, so you cannot really distinguish the hierarchy in these unmutated clones. However, when Ig mutation is present, then most clones early in life show this picture. Also, you see there is a steady decrease over life, and we see more clones that look like this. And there you see that the sequences from blood and spleen are more and more intermingled. We think this points either to increased recirculation from the splenic archive, which would then again mean that this archive becomes leaky in old age, possibly impairing its protective function. Next, we investigated the relation of IgM and class-switched B-cell subsets, while in peripheral blood you can see that increasing number of clone members correlates with mixed clones, so that means that both IgM and class-switch sequences are present. In contrast, in splenic clones, the picture looks completely different. From this, we assume that the spleen not only collects all memory B cells from the circulation, but also expands them in a fashion which avoids class-switch recombination. This process might be important for lymphoma genesis. I think this I'm going to skip. And this is a graphical summary of our findings. While early in life, you see quite heterogeneous picture, both in the splenic marginal zone and also in the splenic white pulp compartment. With age, we see an increased recirculation, and that most of the sequences that were most of the B cells that are present in spleen belong to expanded clones. Some of these clones even reach 0.1 or even 1% of all B cell receptor rearrangements that were analyzed. These results were published recently in the Journal of Experimental Medicine. Finally, there's one piece of unpublished paper that I would like to share with you. In our analysis, we found that similar immune responses in different people elicit similar antibody sequences, which we then call clonotypes. And we quantified these in two different ways. Here you can see an unsupervised analysis, which basically goes by similarity. And the clonotypes were defined as clonally related sequences present in different individuals. And as you can see, there are two phases in life. Early in life, there are these neonatal B-cell clonotypes, which are characterized by the absence of Ig mutations. And later on, we see memory B-cell clonotypes. These are mutated, and moreover, we collected in the literature over 3,400 Ig sequences with known antigen specificity. And when we looked for these sequences that, for example, recognize viruses, bacteria, fungi, toxins, etc., we saw for these selected antigens that, for example, tetanus, there is a quite constant level of B-cell receptor sequence abundance throughout life. But in contrast, influenza or Streptococcus pneumonia follow an age-dependent distribution. And it becomes even more interesting when we quantify the diversity of these sequences, which is basically the abundance multiplied with the number of individual clones. There we see, for example, in old age, over the age of 50, that the diversity for many pathogens is compromised. This is currently ongoing research, but we think that this might play a role in the susceptibility to infection in, for example, old age. Thank you very much for your attention. These are the people who contributed to this research. Do you have any questions?
Of the immunogenetic features of splenic marginal zone lymphoma with your splenic marginal zone B cells. Are these really related from a immunogenetic point of view? Did you look at that?
Unfortunately, we do not have so many splenic marginal zone lymphoma cases in our hands, but I know there was a recent work which was published, I think, in Blood, which shows that a large fraction of splenic marginal zone lymphoma cases is not directly related to splenic B cells, but that there are many cases, for example, on a molecular level. I think the analysis included both RNA sequencing and also epigenetics analysis by at least a TechStack or maybe even additional analysis, which showed that many marginal zone lymphoma cases are also related, for example, with follicular lymphoma or DLBCL. So there's quite a high heterogeneity in this disease entity, and I think it's hard to tell by these constraints.
Is it possible to identify clones in the peripheral blood that were then detected in the spleen many, many years later in the same matched sample?
I think in principle this might be possible. However, the experimental setup might be somewhat complicated. In this case, you cannot really anticipate from which donor you would expect such a sample. I mean, maybe you could do it by using a biopsy in some way, but I think this is not routinely done.